Blood group systems is based on the antigens which are present on the membrane of the RBCs. So these antigens are basically the proteins or the carbohydrates which are present on the membrane of the RBCs. Now the RBC membrane has many types of carbohydrates and many types of proteins, isn't it? So there are different types of blood group systems as well, and depending on the similarity of the structure of these proteins and the carbohydrates, they are grouped into blood group systems. Okay, we have more than 30 blood group systems that have been discovered, and there are many which have not been discovered till now as well, because these are just the antigens which are present on the RBC membrane, right? So, based on whether these antigens can elicit immune response, that we will see a little bit later, that whether the antibodies can be generated and how much they are generated against these antigens, based on that we classify blood group systems into major blood group system and minor blood group systems. And as I told you, there are more than 30 types of blood group systems which have been discovered. Major blood group systems are the ones which have more antigenicity, that is, the ability to elicit an immune response is much more, and these include the ABO blood group system and the RH blood group system. So we will see these ABO and RH blood group systems in much more detail compared to the minor blood group systems. So what is this ABO blood group system? This is basically the antigens which are present on the RBC membrane. So here this diagram is showing like RBC, and this is the antigen. We are talking here about the ABO blood group system. So the antigens which are present in ABO blood group system is H antigen. Okay, and over this antigen, sometimes there is addition of other carbohydrates as well. Okay, so based on that we have A antigen and B antigen. If there is a presence of the gene which adds a component N-acetyl galactosamine, so there will be one moiety added here, and this will be known as A antigen. On the other hand, if there is presence of gene which adds galactose on this, okay, so that will cause the B antigen to be present. So understanding, H antigen forms the backbone over which if this N-acetyl galactosamine is added, that is, the presence of an enzyme is there which transfers this N-acetyl galactosamine over this H antigen backbone, that is known as A antigen. And if galactose is added, that is known as, say suppose galactose is added, that is known as B antigen. And if both are added, then that is known as AB, presence of both A and B antigens, right? So based on this, there is presence of different blood group types in this system, which include A blood group, okay, in which only A is present, antigen is present. B blood group, only B antigen is present. Then there is AB blood group, okay, both A antigens and B antigens are present. Or if both are absent, then it is known as O blood group, fine. So in major blood group system, we have ABO and RH. ABO has 4 types of blood group: A, B, AB and O. RH, again, there are different types of antigens are there. There are 3 major antigens, that is C, D and E. Out of this, we talk mainly about D because it has the maximum antigenicity. Antigenicity, again, that is the ability to elicit the immune response, ability for the generation of the antibodies. Okay, so what happens, this CD antigen, again, see, there will be genes. From these, either these antigens will be produced or there will be genes in which these antigens will not be produced. Okay, so there is a, like everybody has pair of chromosomes, right? So one gene is present on each of these chromosomes. Say suppose the gene is C, so that means C antigen will be present. Okay, then if it is D, that means D antigen is present. Okay, if it is like this DD, so there will be absence of D antigen. So we will talk mainly about this G antigen. If it is present, then it is known as Rh positive blood group, and if it is absent, that is the alleles are like this, small d, then it is known as Rh negative blood group. Okay, so if we combine both these blood group system, because these are major blood group systems, we will have different types of blood groups. Basically 8 types of blood groups will be there, which include A positive, B positive, AB positive, O positive, right? Positive is for the presence of the Rh blood group system as well, Rh antigen as well. And then other 4 will be A negative, B negative, AB negative and O negative. So total we have 8 types of blood groups which are dependent upon these 2 major blood group systems. Apart from these, we have minor blood group systems, as I told you. Minor blood group systems we don't tell for each individual, right? So if we go for that, then each and every antigen of the RBC we have to determine which antigen is present, which antigen is absent. That is not possible, but still we have found some minor blood groups, and these include MN system, okay, Kell system, Duffy system, right, Lutheran system, many are there. These are some names you should remember. So we will discuss the importance of this minor blood group system when we talk about cross matching. Now first we will talk about how these major blood group systems are inherited. Okay, that is from mother and father, how they are inherited by the baby. So first is about the inheritance of ABO blood group system. So in gene, there are 3 different alleles. What is alleles? Allele is basically the variation of the gene, right? So either the gene can be like this, or the gene can be like this, okay, or there can be a gene with neither A nor B. A gene which is IA, that means it will synthesize from the gene, there will be transcription and there will be synthesis of one protein, that is enzyme. So this enzyme is able to transfer N-acetyl galactose amine, okay, on the H antigen. Then there will be expression of A antigen. If the allele is like this, then there will be expression of B antigen, and this IO is a functionless allele. So there will not be any expression of A antigen or B antigen, right? So this is functionless. Now, in the child, there are 2 chromosomes, right? One is coming from the mother and one is coming from the father, right? So this may be from the mother and this may be from the father. Now say suppose on one chromosome we have IA and the other chromosome we have IO. Then will A antigen will be expressed? Yes, A antigen will be expressed, right? On the other hand, if we have IB from the father, then B antigen will also be expressed. So if both are present, both will be expressed. If only one is present, one will be expressed. If it is IO on both, then we will have O blood group system, because neither A nor B will be present. So inheritance of ABO blood group is codominant. Codominant, that means if the allele is present, then that antigen is expressed. If both are present, AB, then it is not like one is more dominant than the others. That means both are equally dominant. So that is why it is known as codominant, and both will be expressed. So based on the combination of these alleles, we have 6 genotypes. What are the genotypes? See, just make the combination from the mother chromosome and from the father chromosome. What will be the combination of the genes? It will be like OO, right? OA, OB. Okay. Similarly, do for others how it will come. AO, AA, AB, right? BA, B, then BO, and finally there can be AB, BA, right? So we have so many types of genotypes, but here I have written 6 genotypes because you see some are common, like this OA and AO are same, right? Similarly here it is from mother, here O is coming from the mother, here O is coming from the father. So in whole we have 6 different types of the genotypes, which include OO, OA, OB, okay, then there is AA, then there is BB, right, and we have AB. So these are the 6 different types of genotypes, and this gives rise to 4 different types of blood groups, not 6. How? See, if both are OO, then there will be O blood group. If one of these or both of them are A, then there will be expression of only A antigen, right? So this will lead to A blood group. This will lead to OB or BB, it will lead to B blood group, and AB will lead to AB blood group, right? So this is inheritance of ABO blood group system from parents to the child. Coming to inheritance of RH blood group system. Inheritance of RH blood group system is dominant. It is not co-dominant. Okay. So all these three are different antigens, and all of these are compulsorily present. So one is coming from mother, one is coming from father. For both of them, for all three of them, C, D and E. So it can be like this: CC, DD, EE. Right? Or it can be like this. Okay. So even one, if it is coming from the mother, then it will be expressed. Okay, or it may be coming from the father. So these antigens will be expressed. If the gene is coming from both the parents such that this gene is not expressed like this, okay, then in that case we will not have the presence of this Rh antigen. So this will be Rh negative. But as I told you that Rh negative is, the name is only based on the presence of D antigen because it is highly antigenic. So we look only for D antigen. Fine. So this is the inheritance of ABO and Rh blood group system. Now till now we have talked only about the presence of the antigens. These antigens are present on the RBC membrane. But when we talk about the RBC antigens, then we also talk about the antibodies which are present in the blood. Okay, so these antibodies which are present in blood, these are known as iso-agglutinins. If these antibodies are present naturally, we will try to make the sense of it. Iso-agglutinins are present in ABO blood group system. What happens? Suppose my blood group is A blood group. Okay, so A antigen is present on the RBC membrane. Now, when the baby is born with the A blood group, he starts eating food from outside, right? And these external products which he is consuming, there is presence of certain antigens which are similar to that of the ABO antigens of the blood group. So when he is exposed to these antigens which are not present in his body, for A antigen, B antigen is not present. So he will be exposed to this B antigen from the food or from the bacteria which is coming into the body, and this will lead to production of antibodies. Okay. So exposure to antigens from food bacteria leads to the production of antibodies approximately 2 to 8 months after birth. Okay. So A antigen, if it is present on the RBCs, there will be production of B antibodies in blood. Okay, A antigen on RBCs, presence of B antibody in blood. And these antibodies which are produced, they are of the IgM type. Remember, IgM type, not IgG type. These are natural antibodies. So that is why they are known as iso-agglutinins, and these are of the IgM type. Right? Now, based on this, Landsteiner gave a law which is known as Landsteiner's law. According to this law, there are two components of the law. One, if antigen is present on RBC, then the corresponding antibody, which is also known as agglutinin, is absent from the blood. So if A antigen is present on RBC membrane, then A antibody is absent from the blood or the plasma. Second component of Landsteiner's law says, if antigen is absent on RBC membrane, then corresponding antibody or agglutinin is present in plasma. Understanding? So for a person with A blood group, A antibody will be absent from the plasma and B antibody will be present in the plasma. This is the Landsteiner's law. But remember that Rh antigen is exception to this law. That means if Rh antigen is present in the blood, then Rh antibody is absent. So this first component is correct. But if Rh antigen is absent on RBC, still there is no antibody present in the plasma. Understanding? Let us see it in a little bit tabular form, then you will understand it better. So what are the different antigens and antibodies which are present in different blood groups? Suppose the blood group is AB positive, then antigen will be A, right? B, and for positive, that is the Rh antigen, antigen will be D, right? What will be the antibodies? No A antibody, antibody B also absent, and antibody D also absent, so no antibodies. Fine. AB positive, AB negative, A, B, and no D antigen. What will be the antibodies? There will not be any antibodies again, because for D antigen again there will be no antibody, because Rh is an exception to second component of the Landis-Steiner's law. A positive, what will be the antigens? A and D. What will be the antibody? B will be the antibody. A negative, similarly you do for all. Let us quickly do A negative, A antigen, B antibody. B positive, B antigen and D antigen, antibody will be A. B negative, B antigen, A antibody. O positive, no antigen. Okay. And what will be the antibody? A and B will be the antibodies. O negative, O positive will add D antigen as well. O negative, we will have no antigens, right? And no antibodies. Fine. So these are the antigens on the RBC membrane and antibodies in the plasma, which are present in different blood groups, the major blood groups. Now, why we want to know about these antigens and antibodies? Because knowledge of this is important for blood transfusion. There might be cases where blood transfusion may be required by a person. If we know what are the antigens and antibodies present in the blood, then we can prevent mismatched blood transfusion. We'll see what is mismatched blood transfusion and all, and why we have to give a particular kind of blood only to a person. So for this, we need to know the blood type or the blood group of the person, right? So what we do, we take a sample of the blood. I will not talk about the whole practical, just principle we will discuss here. We take a sample of the blood. It is anticoagulant, and anti-sera is added from outside. This is not the plasma one, okay? We add outside antibodies. So this is the sample, and we add different antibodies for different antigens of the RBC. So there is anti-A we add, then we take another sample in another slide, we add anti-B, and we add anti-D. And then we look for clumping of the RBCs, okay? Because if antibodies react with the antigens, it will cause all the RBCs to come together, to clump together, what is known as agglutination, okay? So we look for the presence of agglutination, right? So suppose if I give you one example here, say suppose here there is presence of agglutination, RBCs clump together. Here also agglutination, here also agglutination. That means this sample consists of A antigen. A antigen is present, that is why it is reacting with anti-A antibody. It also consists of B antigen and it also consists of D antigen. Now with this, can you tell me the blood group? Yes, see, A and B antigen, that means it is AB, and D antigen also present, so it is positive. So the blood group is AB positive, right? Suppose if here we see no agglutination, clumping doesn't occur, but in the second one we see agglutination, and here again we see no agglutination. So only second one agglutination is present. So what is the blood group? It will be B, because B antigen is present, that is why we are seeing the agglutination, and no A. So here the blood group will be B negative, okay? Now, suppose there is a person who requires blood transfusion. There are many indications of the blood transfusion. Say, suppose the blood loss has occurred, massive blood loss has occurred, and blood transfusion is needed. So now, what are the things which you should do so as to give compatible blood to the person, and why we need to give the compatible blood to the person? We will take one example. Say, suppose the blood group of the person is D positive, right? Now, which blood should I give to the person? Can I take a blood from any person and give it to him? No, it is not possible. Why? Because this person has A antibodies. That is based on the Landis-Steiner's law, as I told you, right? So I have to give a blood such that this A antibodies do not react with the blood. We'll take some examples. Say suppose this is the RBC of the person, of the patient. We will call him recipient, right? And as I said, it is B positive. So B antigen is present and D antigen is present and A antibodies are present. Now suppose a donor is there whose RBC is this. Okay, so this is donor RBCs, and there will be this plasma also, blood we are transfusing, right? So in plasma there will be antibodies. Say suppose I give the person A positive blood. Okay, that means in the donor A antigen is present and antibody B is present. What is going to happen? See, this blood is going to enter into the patient's blood. So this is the blood vessel we gave, and this is going to enter into the patient's blood. And here are the antibodies. Which antibodies are there in this? There will be A antibodies. And you have given a RBC, let us draw of different color, which has A antigen, right? So A antigen, A antibodies, they will react. This will lead to clumping of donor RBCs. Right? So they all will come together like this in the circulation, and they are going to block the blood vessels of the circulation. And then what will happen, that macrophages will come and they will damage these clumped RBCs, these agglutinated RBCs, and there will be hemolysis. So that lot of bilirubin or hemoglobin is released into the circulation, which ultimately will be degraded into bilirubin, metabolized into bilirubin. Okay, so that we don't want, that the donor RBCs get hemolyzed in the recipient's blood. Now the reaction can be other way around also. You see, the donor blood also consists of the antibodies. What are the antibodies? B antibodies are there, and B antibodies are going to react with the recipient's RBCs. Yes, it can react, but it doesn't happen. Recipient RBCs doesn't get hemolyzed. Why? Because we are giving only 350 ml of blood, that is 1 unit of blood in 5 liters of the recipient blood. So these antibodies of the donor's blood get diluted. So they are not effective in agglutinating the RBCs, right? So what is important in this, that the donor RBC should not get hemolyzed. That we don't want, because the donor antibodies are not effective in the recipient's blood. Fine. So this blood typing is very important for blood matching, for blood transfusion. So who can give blood to whom? Let us see. So before blood transfusion, we do the blood matching, and this is just a revision of that what are the antigens and antibodies present. By looking at these antigens and antibodies, let us see that who can give blood to whom. Say suppose we are giving blood to a AB positive person. So AB positive person doesn't has any antibodies. That means recipient's antibodies are not there, right? So they cannot attack RBCs of anybody, no antibodies, right? So anybody can give them blood, even A positive, B positive, O positive, anybody can give them blood since there are no antibodies, isn't it? So these AB positive people, they are known as universal recipients since there is no antibodies in the recipient's blood. On the other hand, for others, let us see, what about A positive person who can give blood? You see, A positive person has B antibodies, so they can attack B antigen. Who is having the B antigen? B positive person and AB positive person. So they cannot give blood. But O positive person doesn't has any antigen, right? So these antibodies will not be able to attack the O RBCs. Okay. So O person can give the blood and A positive person can also give the blood. Similarly for AB positive, we have given 3, AB positive person can also give the blood to them. So for A positive person, anybody who doesn't has B antigen, right, they can give the blood, that is A positive or O positive. For B positive, who can give the blood? Anybody who doesn't has A antigen, because B positive person has A antibody. So who can give the blood? B positive person can give the blood and O positive person can also give the blood. What about O positive person who can give the blood? See, they have antibodies, both antibodies A and B, so they will attack all these blood. So only a O positive person can give blood to a O positive person, right? So remember the concept of universal recipient. AB positive person is a universal recipient because he does not has any antibodies to attack any donor RBCs. On the other hand, O person is a universal donor. Actually, O negative is a universal donor. Why? Because we don't have any antigens on O negative person. So no antibodies can attack this O negative person. So that is about the blood typing and blood matching. But before blood transfusion, we also do cross matching. What is cross matching? I told you in the beginning about the minor blood group antigens. Yes, minor blood group antigens have very less antigenicity, very less probability of causing antigen-antibody reaction. But still they may occur, right? So for that, cross matching is done. And what is cross matching? We have major and minor cross matching, out of which major is the important one. So what we do, we take recipient's plasma, okay, recipient's plasma, and then we mix it with donor RBCs. So in plasma antibodies are there. So it is like in general, if anybody's antibodies are present that can react with the donor RBCs, then there will be clumping. But remember, first we will match, okay, otherwise ABO mismatching, definitely there will be clumping. So first we match, then matched blood we take, and then we do this cross matching. And if any antibodies against any minor blood group antigens, they are able to react with these donor RBCs, then we go for another ABO blood group matched blood, okay. So this cross matching is to detect antibodies against minor blood group systems, antibodies against minor blood group systems, right? Then minor cross matching, we do opposite, that is recipients RBCs plus donor plasma. Do you think it will be important? No, not important, because again donor antibodies will get diluted in the recipient's blood. Okay, so cross matching is also done before the blood transfusion. Coming to applied aspects. So first very important one is mismatched blood transfusion. Whenever blood transfusion is needed, we have to make sure that it is the matched blood groups. Okay, then only the blood transfusion is done. Otherwise, there will be agglutination of the donor RBCs, and this ultimately will lead to delayed hemolysis. That means all the RBCs will clump. Then the macrophages are going to attack these RBCs for phagocytosis. There will be damage to the membrane of these clumped RBCs. Hemoglobin will be released into the circulation. Or there can be immediate hemolysis also if there is huge number of antibodies which are present. Okay. And all this hemolysis will ultimately lead to renal failure, because lot of damage to RBCs, it can lead to circulatory shock. It also causes the release of toxic substances which leads to vasoconstriction in the renal vessels. Okay, so renal vasoconstriction, thus it decreases the blood supply to the kidneys. It also leads to hemoglobin filtering into the nephrons, and ultimately it gets precipitated in the nephrons. Okay, so this leads to tubular necrosis and ultimately renal failure, and death of the patient can also occur. So it is very important that only matched blood is transfused to the patient. And in this there is a concept of autologous transfusion, that if we are doing a planned surgery, then in that case over a period of 3 weeks blood is collected from the patient himself. Okay, so 2-3 units of the blood can be stored depending on the requirement, and it is done over a period of time, so that not all the blood is taken out in one time. 1 unit of blood is first taken out, then iron therapy is given so that the RBCs are synthesized and released into the circulation. Then after some time again some blood is taken out, so the blood is stored, and then the same blood, the patient's blood only, is transfused to the patient if required in the surgery. Okay, so that is known as autologous transfusion. Coming to second important applied aspect, that is erythroblastosis fetalis. What is erythroblastosis fetalis? It is a disease of the fetus and newborn. It occurs if Rh negative mother bears a Rh positive baby. And when it can happen? When the father is Rh positive. As I told you about the inheritance, if the alleles are either this, okay, both D antigens, both the chromosome of the father is having D antigen, or even one if it is D antigen, then in the first case, definitely one of these is going to go to the baby, right? And Rh negative mother means it is like this, right? So one gene is coming from the father, that is definite cause Rh positive baby, and in other case there are 50% chances that the baby will be Rh positive. So in this case, what happens in the first pregnancy, say suppose this is the fetus, right, and this is the mother. The maternal circulation has RBCs which are D antigen negative, and there will not be presence of any antibodies, right? So no antibodies in mother, but in the fetus there is Rh positive blood. Fine, in the first pregnancy it will be like that, but during delivery what will happen, that some of this Rh positive blood of the fetus will enter into the maternal circulation. And because of the exposure of this Rh positive blood in the maternal circulation, there will be generation of antibodies. Okay, so this IgM antibodies are being now synthesized in the maternal blood. But now the fetus has been delivered, so nothing has happened to the fetus in the first pregnancy. But in second pregnancy, what happens that again, if the fetus is Rh positive, then during pregnancy also small blood leaks out. Okay, so this ultimately will lead to the formation of IgG antibody, and this IgG antibody is going to cross the placenta, enter into the fetal blood and destroy fetal RBCs. So there will be hemolysis of the fetal RBCs in the second pregnancy and subsequent pregnancies. Okay, so this is dangerous. What will be the features which will occur? Because of the hemolysis of the fetal RBCs, there will be anemia. RBCs are being destroyed. There will be jaundice, right? And there will be edema. Okay. Then, because of so much destruction of the RBCs, the fetal body will try to synthesize more RBCs, and that will happen in liver and spleen. So they will enlarge. Then, whenever there is increased synthesis of RBCs, even immature RBCs are released into the circulation. So immature blast cells are released into the circulation. See, the name of the disease is erythroblastosis fetalis, right? Blastosis, that is immature blast cells, right? They are released into the circulation. Then, since so many RBCs are being destroyed and there will be increased formation of the bilirubin, so that is going to lead to jaundice. Plus, what happens that the fetal blood-brain barrier, that is immature, okay. So this bilirubin can cross the blood-brain barrier and it can enter into the brain and get deposited in basal ganglia. Basal ganglia is important for motor movements, right? So this leads to abnormality in motor movements, right? So this is dangerous because once this damage occurs, it cannot be reversed. Then because edema occurs, this condition is also known as hydrops fetalis. Hydrops fetalis. Then there is another name for this condition, that is hemolytic disease of the newborn. So erythroblastosis fetalis, hydrops fetalis, hemolytic disease of the newborn, all three are same things, and these are the features of the condition. Also, if this hemolysis is severe, there can be death of the baby also, right? So how to prevent this disease? So for prevention, we give one dose of anti-D antibody to the mother within 72 hours of delivery or parturition. Okay, so when we give this anti-D antibody to the mother within 72 hours of delivery, the fetal RBCs which have entered into the circulation, say suppose these are the fetal RBCs, this anti-D antibody goes and binds to the D antigen which are present on the fetal RBCs. Okay, so it is covering those D antigen. Hence, the maternal blood is not getting exposed to this D antigen. So there will not be any production of antibodies in the mother's blood, right? So it is preventing the sensitization of the mother's blood. So that is the prevention by giving anti-D antibodies to the mother within 72 hours of delivery. But suppose that the baby gets affected, then we have to treat the baby, right? So the treatment for this is exchange transfusion. See, the Rh antibodies from the mother are destroying the fetal RBCs. So we have to prevent it. So after the baby is delivered, we take out the Rh positive blood of the fetus and transfuse the Rh negative blood. Understanding? So this Rh negative blood doesn't has Rh antigen. So the antibodies which are directed against the Rh antigen, they will not be able to act on this. They will not be able to destroy it. Okay. So this exchange transfusion, removal of the Rh positive blood and transfusing Rh negative blood is done quite a few times in first 2 months after delivery. And over time, the antibodies which have entered into the fetal blood, they will also be destroyed. So over time, when these antibodies are removed, then this exchange transfusion is stopped. Fine. So that was all about the blood group systems, the major blood group systems, minor blood group systems. What is the importance of these two different types of blood group systems, how they are inherited, what is isoagglutinins. Then we also discussed about the blood typing, blood matching. We discussed about the blood transfusion and what if mismatched blood transfusion is done, and we also discussed about the erythroblastosis fetalis, a very important topic for undergraduates. Thanks for watching the video. If you liked it, do press the like button, share the video with others, and don't forget to subscribe to the channel Physiology Open. Thank you.